In the year 2022, a Taiwanese young adult who just got his master's degree passed away in his apartment because of the side effects from COVID vaccination. The vaccination clogged out his blood vessels, and he was killed by pulmonary embolism. During the time of COVID, we faced many challenges and heard countless horrifying stories. What happened to this young adult is definitely not the first case, and for sure won't be the last as well. The vaccines were supposed to save lives, yet it has the devastating side effect of causing thrombus. We're a team wearing Out Clots from Taiwan, and thrombus has successfully caught our attention.
Due to COVID, the issue of blood clots has been an immediate public concern. After some research, our team decided to dive deeper into cardiovascular diseases, otherwise known as CVDs. CVDs are among the top 3 causes of deaths globally, with a prevalence of 1 in 13 individuals. And they claim approximately 20.5 million lives while being responsible for 40.8 million disability-adjusted life years annually around the world. Economic costs associated with CVDs are approximately 282 billion euros annually. As we can see, CVDs are not only a ticking bomb in people's lives, but also a huge burden to our healthcare system. After researching worldwide about CVDs, we decided to bring our focus to our home, Taiwan. Taiwan faces an even more serious problem when it comes to CVDs, as we are facing an aging population crisis. The elderly population has been growing exponentially over the years, with percentage of the population aged 65 and above at 13.2% in 2016 and projections of a 19.8% increase by 2025. We can also see from this that CVD's prevalence rate is higher in the elderly population. In 2017 alone, CVDs such as heart valve disease had a cumulative incidence rate of 3.59%. while also possessing a higher incidence of a surgical valve replacement at 6.99% in individuals aged 65 and above. Through this research, we noticed that a lot of our senior citizens have died from CVDs. Starting from this issue at hand, we began to brainstorm possible solutions to resolving issues caused by aging populations and CVDs. Of course, there isn't just a single variable in the equation of CVDs, so we focused our attention on the thrombus, setting it as our main research target. In order to find a breaking point for the thrombus, we broke down the causes of thrombus into 3, including endothelial injury, slowed blood circulation, and abnormal blood composition. Endothelial injury is mainly caused by high blood fat content and hypertension, which could cause LDL cholesterol invasion and cause atherosclerotic plaque. Then, when blood pressure skyrockets or blood flows abnormally, plaque will crack and produce white thrombus. Strokes are also a common example. Blood flow disorder is usually caused by atrial fibrillation, mostly in veins, which end up causing red thrombus, and abnormal blood content may cause high blood viscosity, causing deep vein thrombus, which is also a kind of red thrombus. Regardless of the type of thrombus, its composition is mainly fibrin. We figured that the breakthrough point might just be fibrin, so we decided to find a solution to dissolving fibrin.
Through the research of vast amount of academic papers regarding dissolving fibrin, a word kept showing up, and it was called lumbrokinase. Lumbrokinase is an organic compound found in the body of worms. When the worms die, the lumbrokinase they possess in their bodies works to dissolve their own corpse, returning them to the mother nature. Scientists further experiment on this trait, finding the enzyme to hold the ability to dissolve fibrin. So we decided to use lumbrokinase as our weapon to defeat the evil fibrin. Lumbrokinase has 3 advantages: safety, biological possibility, and potential for development. First, safety. In 2022, the current lumbrokinase supplement on the market has been approved by Monde Selection and SNQ. Second, biological possibility. Academic papers have mentioned that laboratory experiments and clinical experiments have been done to support the possibility of lumbrokinase in dissolving fibrin. Thirdly, the potential of development. Lumbrokinase as a supplement have more room for improvement and overall potential for our high school team compared with the medical drugs.
To further understand lumbrokinase, we interviewed Darwin Vermiculture Limited Company's boss and learned that majority of lumbrokinase on the market comes from worm juice. Worms are shredded for their juice, and this way of production of lumbrokinase is not only inhumane but also costly. In addition, the lumbrokinase that is produced is not of high quality. Therefore, our project aims to find a solution to increase the effectiveness of the industrial solution. Then, in order to receive more information on CVDs, we interviewed Dr. Lin from Chang'an Hospital. He mentioned that the main reason for CVDs among Taiwanese citizens are their high-fat diets, and elderly often forget to take their pills, and it worsens their CVD symptoms. This inspired us to build a probiotic platform to address the problem of elderly forgetting to take their pills and their high-fat diets. But firstly, after fully understanding the research directions and the stakeholders, we have to begin designing our industrial production.
At the very beginning of the experimental design, we had to obtain a lumbrokinase gene sequence and ligate it with the pGMT-easy vector that we designed. In order to achieve the first step, we need to find a gene sequence in worms that produce lumbrokinase and modify it. However, it didn't go as smoothly as we planned. In the experiment, we adjusted the diet of the worms by feeding them the ionized water on the tissue and dehydrating them,
so that their stomachs would be less contaminated by microbes from their regular diet of soil and dirt. Our adjustments result in successful RNA extraction from the worms, and we use steps like reverse transcription and gel purification to obtain the lumbrokinase gene sequence while connecting it to the pGMT-easy vector. This was a huge breakthrough, as we extracted lumbrokinase RNA, the Taiwanese earthworm, instead of the Japanese one documented in the research.
Now, we can see that the pGMT-easy vector has a 99% sequence alignment, showing that our vector contains the correct lumbrokinase gene sequence. Mentioned previously, the traditional way of extracting lumbrokinase is human, costly, and not effective. Therefore, we hope to use IPTG induction to increase the effectiveness of industrial production. After we obtain the correct system, we use PET22B vector for gene expression to meet the requirements of industrial production. The PET22B vector has a special mechanism called IPTG induction, which works by using allolactose to induce transcription. This is the first step to protein synthesis. The lactose upron in PET22B vector is the kill switch for the whole induction.
After using IPTG induction to produce lumbrokinase and meeting the required rate of industrial production, we started brainstorming how we can use lumbrokinase in the probiotic platform. As previously mentioned, when our blood pressure skyrockets or blood flow is disorderly, plaque builds up and forms the white thrombus. High-fat diets are one of these causes. Therefore, we decided to use oleate, a type of fatty acid, as an induction molecule to make sure that our probiotic platform can produce lumbrokinase after detecting a certain amount of fatty acid in our body. Then, lumbrokinase can be used to dissolve fibrin and solve plaque caused thrombus.
We decided to modify PET22B vector and change the lactose upron to fat R binding site, otherwise known as fat edge operator. In our experiment, we used oleic acid as an induction material to ensure that after detecting a certain concentration of fatty acids, our E. coli will start producing lumbrokinase. After analyzing the results from our gel, we found out that oleate-induced PET22B vector is the same as the one we used for IPTG induction, proving the plausibility of gene editing. After the completion of our plasmids, we used transformation to E. coli BL21 for protein expression. We also tested the induction mechanism of oleate and IPTG. First, we used Western blot to test the results of IPTG induction. We can see that the concentration of lumbrokinase peaked around the 6-hour mark, proving the effectiveness and plausibility of IPTG induction. Then we used Western blot to test oleate induction. Different from IPTG induction results, the concentration from oleate induction peaked around 24 hours. In addition, we used different concentration of oleate induction and analyzed lumbrokinase production results. In this image, we can see that the three concentrations of oleate induced the same amount of lumbrokinase. Although both results point to oleate induction being less successful compared to IPTG induction, there is still enough evidence to prove the plausibility and effectiveness of oleate induction. To conclude, our two Western blot results supported IPTG and oleate induction being successful in producing lumbrokinase, showing the plausibility of our experiment.
Our experiment was a success. Of course, our project didn't stop in the lab. To further introduce this innovative way of lumbrokinase production to supplements, we have to go through different tests and concerns in order to turn theory into application. We decided to interview the Vice President from Medigen Vaccine Biologics Corporation, Dr. Lin. During the time of COVID, he led Taiwanese scientists and doctors to develop a COVID vaccine. During our interview, Dr. Lin mentioned that if we wish to turn lumbrokinase into a medical drug, we are going to have to go through a series of in-vivo experiments. That's when we realized that our project will face many challenges in the future, but is also full of promise and potential. After valuable insight from Dr. Lin, we know if we want to be more influential, we need to commercialize our product. We decided to interview a key figure of the supplement market, CEO of BioKing Health Technology, Mr. Luo, and lumbrokinase is one of the products under his company. During our interview, we understood that although lumbrokinase has a broad range of applications and users, it's still in an awkward spot between medication and supplements. This dilemma has caused much confusion amongst its consumers, and therefore, we are able to test the plausibility of our probiotic platform, and we can not only solve CVDs but also help with confusion amongst the consumers and promote our probiotic platform to more people in need. After getting a different perspective, we hope to get the views from our peers. Who knows? We might be able to discover something new. Our team was a host for Taiwan's 2023 in-person IGEM seminar. And through this opportunity, we had the chance to compete and communicate with other high school and college teams. This not only helped us locate our weaknesses, but also promotes and encourages future synthetic biology collaborations and projects. After collaboration and our seminar, there was an information exchange that allowed us to learn and receive feedback from different perspectives. So we decided to plan our next steps. This includes regular education programs to spread the knowledge of synthetic biology and to plan the 2024 Taiwan IGEM seminar to unite IGEM participants from all over Taiwan to exchange experiences. To conclude, although our project experiment was successful, we will continue to work on our probiotic platform, especially as it is still in the in-vivo phase. Therefore, we wish to make our in-vivo experiment a reality and work with biotech companies to research the plausibility of the effectiveness of the probiotic platforms in the human body. So, how does our project contribute to our community? First, our project is efficient. Through IPTG induction, we are able to improve the traditional way of lumbrokinase production and greatly increase the production quality of lumbrokinase. Second, our project is up to date. By combining oleate induction and an E. coli platform, we are able to detect and absorb fatty acid when people are on a high-fat diet, produce lumbrokinase to dissolve fibrin. Third, our project is future-proof. Lumbrokinase has completed phase 2 clinical trials, and the effectiveness of lumbrokinase is widely recognized on the market. We are able to ensure the effectiveness of lumbrokinase, creating more possibilities for our project. Lastly, let's return to our original motive. After the results came out and the project was finalized, the problem we wish to address is still CVDs, because this is not only a local problem but also an international one. We wish more people could see our project, spreading awareness and encouraging more projects and research to help the CVDs community. Thank you to everyone who has helped us complete our project, and see you guys in Paris. We are Out Clots.